Okay, so we start the fun, and since we are a bit late with the schedule, I made a scroll to describe our first plenary speaker, but I will make it short. Our first speaker of today, the plenary session, is Professor Itamar Willner from the Hebrew University. Now, we all call him Itamar, but you should know that Itamar is one of the leaders, global leaders in nanotechnology, nanobiotechnology, and specifically dwelling into the electrochemistry of biological systems. Itamar published more than 1,200 papers and was authors of many others, 1,200, more than that. He has close or more than 100,000 citations. Itamar is a global leader in science and technology, and everybody is looking up to him. So Itamar, without any ado, please join us and deliver the plenary talk, and we are all honored by your presence.
Let me thank Marachino Ket and the organizing committee for the invitation to present this first lecture. And my lecture will deal with nanomaterial-functionalized electrodes for bioelectronic and photobioelectronic applications. Now, if we look at biomaterials and nanomaterials, they are more or less of similar dimensions, yet of very different properties. Biomaterials have unique recognition catalytic properties, and nanomaterials have unique electronic, photonic, optical properties. And the integration of these two materials into hybrid systems leads to new functional materials that combine the properties of the two entities. And what we basically established the field of nanobioscience. Now, in this system, the biological functions are reflected in the functions of the nanobiomaterials, and the functions of the nanobiomaterials are reflected in the functions of the biological entity. Now, I would say that during the years I had the privilege to serve and advance this field of nanobioscience, and within these activities there is a niche of an interesting area where biomaterials and bio-nanomaterials are integrated with electrode support, and this establishes basically the field of electrochemical nanobioscience. And today I will discuss some of our advances in this area. And I will try to address three topics. One topic will address the integration of biomaterials with electrode surfaces, and this will be enzymes and nucleic acid. Then I will move on to the integration of redox active hydrogens with electrode support and what can be done with these systems. And finally, I would like to address some aspects of photobioelectrochemical and photobiofuel cell electrodes that combine biomaterials with electrode support. Now, at early stages of our activities, we addressed this topic, and probably many people here in the audience that were my previous students know well that we were trying to understand the electrical contacting of redox enzymes with electrode support, and you realize that when you place an enzyme on an electrode, usually the systems lack any communication, and there is no dialogue between the electrode and the enzyme. And one has to address the difficulties to overcome in order to contact the redox center of the enzyme with the electrode, and we do it basically on the so-called Marcus theory that tries to understand this problem of electrical contacting by bringing together the redox center and the electrode in close distance. And at early stages of our activities in this field, we tried to introduce gold nanoparticles as a particle to contact the redox center of an enzyme and the electrode. And we did it by the so-called reconstitution process, whereby we took an enzyme, we extruded from its active sites, for example, the flavin, adenine dinucleotide phosphate. We generate an enzyme with a gold and apo enzyme, and we implant into it a gold nanoparticle that is functionalized with an FAD cofactor, so that this gold nanoparticle acts as a nanoelectrode to contact the enzyme with the electrode. You can see here a very nice TEM image of the gold nanoparticle that is inside the enzyme glucose oxidase. And as you can expect, once we attached it to the electrode, we had very nice electrical contact between the redox center of the enzyme and the electrode, and we were able to catalyze by means of the system the oxidation of glucose. Once we had this discovery of nanomaterials that can contact with an electrode, we moved on to carbon nanotubes, and we were able to contact the redox center of an enzyme by means of carbon nanotube as a nanoconnector that connects the FAD center with the electrode. And it was accomplished by assembly of carbon nanotubes on an electrode, modification of the head of the carbon nanotube with the FAD cofactor, the semi-synthetic FAD cofactor, reconstitution of the enzyme on the FAD cofactor, and once we had it as a function of length of different carbon nanotube lengths, we were able to contact the enzyme with the electrode and to get the bioelectrocatalyzed oxidation of glucose. Here you can see very nice the standing carbon nanotubes on this electrode before the reconstitution. You can see after reconstitution the enzymes that were on the surface. And finally, we were able to see at the single carbon nanotube level, we were able to see the glucose oxidase units that are attached to the heads of this carbon nanotube after the reconstitution process. These systems are nice systems by demonstrating that nanomaterials like carbon nanotubes or gold nanoparticles can contact redox center with electrodes, but these are basically systems that have an academic significance. The question is how can one scale up the system in order to get useful devices. And this was achieved by taking porous carbon nanoparticles and using the pores of the nanoparticles as a container of redox active components. For example, we immobilized into these pores of the carbon nanoparticles either ferrocene units or methylene blue units, and we kept the pores with enzymes, glucose oxidase or horseradish peroxidase. And once we kept these pores, we got entities that first of all are integrated, totally integrated, contain a high content of these enzymes and the relay units, and they are extremely well electrically contacted. If we apply on this mass of carbon nanoparticle a potential, you can see that we can effectively oxidize by the ferrocene, mediate the oxidation of glucose by the ferrocene embedded in these pores, or reduce with the methylene blue hydrogen peroxide by the horseradish peroxidase enzyme. And these electrodes are stable for weeks and months without any leakage of the components or the activation of the system. Once we had this as a potential use of sensors, and we were able to contact this, the enzymes with the bulk electrode surface, we applied this system and this concept to develop biofuel cells. And in fact, about 10 years before we did this system, I think that we were the first one to generate a biofuel cell that was very inefficient, but it demonstrated the feasibility of a biofuel cell. But once we had this system of porous carbon nanoparticles, we were able to construct a biofuel cell that demonstrates substantially higher activity. And what we did is, as an anode, we used electrically contacted porous carbon nanoparticles that included the ferrocene as an electron mediator, and this system was kept either with lactate oxidase or glucose oxidase as the bioelectronic catalytic active enzyme. And as a cathode, we applied the bilirubin oxidase, and we were able to introduce this ABTS electron mediator into the course of the nanoparticles and kept them in order to get an electrocatalytic electron for oxygen reduction. So we have here the possibility to make with lactic acid a biofuel cell, and with glucose as fuel a second biofuel cell, or alternatively to take glucose and lactate as fuel products, and these are abundant materials, to generate electrical power. And this is exactly what we did. You can see here that the electrocatalytic activity of the anodes and of the cathode. And once we had this characterized separated electrodes, we combined them to generate a biofuel cell. And to see here, you can see here the power output of this biofuel cell when we have lactate as fuel, glucose as fuel, and if we add both systems, we get a substantially higher output that combines the performance of the two elements. So this story briefly summarizes activities to electrically contact enzymes with electrode support and show you what you can do with this type of biomaterial. But I think that one area that we are very much interested is the area of nucleic acid and how to integrate nucleic acid with electrodes in order to get an electrical output or an electrical contact is really a challenge. And those that are familiar a little bit to the field of nucleic acid will realize that we can take a single-strand nucleic acid and in the presence of an appropriate trigger, we can self-assemble very interesting structures of nucleic acid. One of these nanostructures is the structure of a G quadruplex. A G quadruplex is generated by guanosine-rich strands that in the presence of potassium ions self-assemble into a G quadruplex. So what we were able to show, we were able to show that we can take a single-stranded thiolated nucleic acid reaching guanosine bases in the presence of potassium ions. This is this surface, it self-assembles into a G quadruplex, and this G quadruplex binds various transition metal complexes like the imine, and once we bind the imine, we get an electrocatalyst for the reduction of hydrogen peroxide to water. When we add to this system crown ether, we are able to separate, by taking out the potassium ions, we separate the G quadruplets and we get a random coil. So we can switch on and off the bioelectrocatalytic activities of this enzyme elements. But once we have this, we can basically develop a whole series of functionalized electrodes that can act for sensing applications. For example, you can take a hairpin structure of nucleic acid that includes here a recognition sequence for genes, microRNA, and other nucleic acid. And in the stem region, the blue part, we encode the information of a G quadruplex. Once we open this hairpin structure, we generate the G quadruplex, they incorporate the imine structure, and the electrocatalyzed reduction of hydrogen peroxide provides a signal to read out the sensing process and to amplify the sensing event of the respective gene or microRNA. This concept was extended to several other analytes. For example, you have nucleic acid that recognize low molecular weight substances like cocaine, ATP, or proteins like thrombin, and these nucleic acids are called aptamers. So what we are doing, we encode into the hairpin structure the recognition sequence of the aptamer, in this case it is AMP, and in the stem region the information for the G quadruplex. And when we have the analyte, we have the opening of the system, and we catalyze the reduction of hydrogen peroxide as a transaction event for the sensing event. And you can switch it on and off this and regenerate the electrode by dissociation of the G quadruplex and elimination of the guest molecule in the aptamer unit. Till now, I showed you how we can do it with amperometric responses of the electrode, but in reality, you can do it also by impedance spectroscopy, whereby you examine the interfacial electron transfer properties upon transforming the electrodes from the hairpin structure to the G quadruplex. For example, using ferrocyanide as redox reagent, the Faradaic impedance spectrum is strongly dependent on the structure of this nucleic acid functional electrode. In the presence of the slightly positively charged potassium ion stabilized G quadruplex, the electron transfer resistance is substantially lower, and we can sense the event by probing the impedance spectrum of the system. We can use this control of the interfacial electron transfer properties also to catalyze different catalytic transformations. For example, silver clusters or silver nanoparticles that are negatively charged because of the capping of the silver cluster. When we have a normal strength negatively charged nucleic acid, then these catalytic nanoparticles are repelled from the surface. But when we generate the G quadruplex, we allow electrical contact between the nanoparticles and the electrode that will catalyze effectively the reduction, for example, in this case of hydrogen peroxide. And again, in the presence of ground ether, we can switch on and off the catalytic functions of the. But this story is the story of biomaterials on the electrode, and I think that the challenge is to try to develop nanomaterials that have bulk properties on the electrode, and this was accomplished by the deposition of hydrogels on the electrode. It is something that is not very easy. And hydrogels are matrices, soft matrices that are rich in water, contain a high amount of water, and we are not interested to place just hydrogels on surfaces. We are interested to put smart hydrogels or stimuli responsive hydrogels on electrodes. And what I would like to show you, first of all, how one can build up a very thin film of a redox active stimuli responsive hydrogels on electrodes, and how we can apply it for certain applications, especially catalytic and nanobiomedical application, and to try at the end to see whether we can combine this story of nucleic acid, the interactions of nucleic acid with electrodes and enzymes with electrodes, in order to make biomolecular loaded hydrogel that responds to an electrical signal. And I will try to show you at the end that eventually one can try to make an artificial pancreas by this method. Now, the way to put it, and it is not easy to place a thin film of hydrogel on an electrode, and I think that here we have developed a very unique method. This is a versatile and generic method that is based on the so-called hybridization chain reaction. Now, the hybridization chain reaction is a process where we take a promoter unit, a promoter nucleic acid, two hairpin structure. The promoter opens hairpin A. Once we open hairpin A, the open stem region opens hairpin B, and the open hairpin HB opens back HA, so we can generate long polymer chain of hydrides that contain this duplex nucleic acid. This concept was developed by Pierce and Cartet, and we adopted it to develop the deposition method to deposit hydrogen on electrode surface. And here I want to show you the first, the concept. The concept is to immobilize on an electrode surface, for example, a gold surface, to immobilize a promoter unit, to take polymer chain of acrylamide, carboxylated cellulose, a monomer that is modified with two hairpins of DNA. Once we interact these two types of chain with the promoter, we induce the hybridization chain reaction that I just described, and we get a duplex bridged hydrogens on the electrode. This is a film that is on the electrode. This is not yet a smart film. To convert it into a smart film, we attach to the polymer chain additional nucleic acids that include an element of information to be smart. For example, once we take this polymer, we may induce the hybridization chain reaction, and we make the hydrogen on the surface. If this tether as one is enriched, we know already that one is enriched will form a G quadruplex. We will cross-link in the presence of potassium ions the surface not only with the duplexes, but also with the potassium G quadruplexes. And then we get a relatively stiff hydrogen. If we add crown ether, we can separate the G quadruplex and get a hydrogen of lower stiffness. So this is the method to control the stiffness of a hydrogen interface on an electrode support. You can see here, for example, microindentation experiments that really measure the Young modulus of the hydrogen in the presence of without the G quadruplex, with the G quadruplex, back and forth, you can switch it. Once you have this hydrogen on the electrode and control the stiffness of the hydrogen, you are able to do one step further and to make a switchable electrocatalytic electrode. You can incorporate into this G quadruplex the hemine catalyst, and then we can switch on and off the electrocatalytic properties of this electrode, for example, towards the reduction of hydrogen peroxide. So this is a very simple example of trying to make a smart hydrogen surface on the electrode. And from here I will move to a slightly more difficult and more complex system where we try to do this artificial puncture. And the way to do it is basically exemplified here. What we want to make is a pH responsive hydrogel on the electrode. So we make a stiff hydrogel in one case, and under acidic pH, we make a lower stiffness hydrogel with the aim to release at the end insulin. Now, in order to do this, we can certainly construct our hydrogel, and as before, you can see here, we take two polymer chains that include now cytosine-rich nucleic acid. We make duplexes by the hybridization chain reaction, but we add new duplexes of these tethers that are shown here. These duplexes, one of these duplexes is rich in cytosine at acidic pH of 6, 5.5. It reorganizes, reconfigures to the hydrogen structure. You lose a cross-linking element, and you make the hydrogen less stiff. Now, we can do it manually by changing manually the pH, but we can make it in a more sophisticated way to immobilize into this hydrogen glucose oxidase. The glucose oxidase oxidizes glucose to gluconic acid. Gluconic acid generates an acidic environment in the hydrogen, opens the hydrogen, and then as a function of the concentration of the glucose, we will release from this hydrogen our insulin. So this was the concept, and now I want to show you that we can go ahead and have it. First of all, the microindentation properties show you very nicely that in the presence of glucose, the stiffness of the hydrogen is controlled by the concentration of glucose. As the concentration of glucose is higher, we make a higher acidic pH in our hydrogen, and the hydrogen becomes less stiff and of higher capacity and efficacy to release in the future the glucose, the insulin. Now, we were able to follow these permeation properties and pH properties changes in the hydrogen, either by impedance spectroscopy or by really color changes of the hydrogen. And here basically you can see that when we have a very the stiffer hydrogen, neutral pH, we have small pores, dense pores in the hydrogen. When we have a slightly acidic pH, we have bigger pores and less dense pores that can release eventually the load in the future. And here you can see the release properties of this hydrogen, and everything works as expected. We can release either a dye as a model for a load, and we did it with the fluorescein modified dextran, or we can really put an insulin inside that is labeled with a fluorophore and release the insulin from our system. You can see here that the release of the insulin is controlled as expected by the concentration of glucose. So until now, we did everything manually. The electrochemistry didn't enter into our story. And we have now to go to the next step and to convert this entire change of the pH by means of electrochemistry. If you recall from the first slides that I introduced in my talk, we had the oxidation of glucose using a relay unit to make gluconic acid electrochemistry. So if we can make, in the presence of a relay unit, we can make gluconic acid, we can induce at the electrode surface and decrease in the pH and open our hydrogen. So this was exactly the method that we applied, and what we did is we added to the system ferrocene, and you know already that ferrocene, carboxylic acid, is a mediator to mediate the oxidation of glucose to gluconic acid, to reduce the pH, to change the configuration of the hydrogel into the hydrogen structure. And as the concentration of glucose is higher, the degree of pH changes will be higher by the electrochemical method, and we will release a higher content of glucose. And this is exactly what we did. You can see here, first of all, the electrocatalytic oxidation of glucose in this hydrogel. You can see the release of tetramethylrhodamine as a model for insulin, and you can see that the difference, we can switch on and off the release of insulin by applying the potential and switching off the potential on the electrode. There is here one problem in this system. The problem is that our ferrocene is diffusional and has no practical use, so what we did is we attached to the glucose oxidase ferrocene units, and these ferrocene units generated now an integrated system for the switchable release of insulin from the cell. You give me 2 minutes, 3 minutes. In the 3 minutes, we'll address the final topic that I wanted to discuss, and these are photobioelectrochemical and photobiofuel cells that apply elements of nanotechnology. Now, the contacting of enzymes with electrodes introduced the same problem to contact photosystems or photoactive proteins with electrodes. And those that are familiar to the photosynthetic reaction center, the photosynthetic reaction center operates in 2 photosystems, photosystem 1 and photosystem 2. We excite the chlorophyll units in the 2 photosystems, and in photosystem 1, we reduce NADP, and at the end reduce carbon dioxide to sugars. In photosystem 2, we oxidize water to oxygen and complete the cycle. Now, I think that Danny Mandler is well familiar to these schemes since he was doing his work in metabology. In any case, our idea was to try to take from nature, from the plants, from the leaves, to take these photosystems and to make the photoelectrochemical systems. And in an old study, what we did is we took photosystem 1, we contact the photosystem 1 with a phenol polymer as an electron relay with the electrode, and we get here the photoelectrochemical electron transfer to one electrode, and on the counter electrode we have the reduction. We contact bilirubin oxidase to reduce oxygen to water, so we generate a photoelectrochemical cell that generates photocurrent upon irradiation. This is a very simple method that I don't want to discuss it in details, but it includes a lot of elements of nanotechnology. For example, we were able to couple also photosystem 2 with an electrode support, and generate a current out of a photocurrent out of this system. But the major advance came when we were able to really construct the two photosystems, like in nature, to make an entire Z scheme. And what we were able to do is to construct the photosystem 1 on an electrode support, photosystem 2 in between them to make a polymer layer with cytochrome C like in nature. Photosystem 1 is contacted with the electrode support by means of in this case, the platinum nanoparticles, like in the first slide, it was the gold nanoparticle. The oxidized center oxidizes here cytochrome C, and photosystem 2 is contacted by means of the oxidized cytochrome C that is generated in between the two photons. This is a way to model really photosynthesis, but the question is, can we take all the stories that I told you and integrate it to one system? And the system would be a system where we contact by means of a photosystem a bioelectrocatalytic transformation. And this will be the oxidation of glucose. And this is exactly what we did. We had a system, we constructed a system that is based on photosystem 1, a relay unit that contacts this photosystem 1 with an electrode, and we oxidize here a second relay unit, R2. This relay unit oxidizes the enzyme glucose oxidase, and we have the oxidation of glucose, so we have the fuel substance is glucose, and we convert this fuel product into an electrical power that comes out of the system. You can see here the combination of this electrode. We have the relay is a so-called pyrrolophenolinoquinone, but we have another one that you will see soon. We contact it with the enzyme by means of a polymer that is a redox active polymer. All the components have the appropriate redox level to be electrically contacted one with another. We can follow the entire step of combination of these electrodes. By the way, this was published recently in Nature Energy, this system, so you can have the details there. And we were able to get out of this system, the electrical power upon oxidation of glucose. Now this is a lesson that teaches us how to do everything in a synthetic way. I will jump over the two slides that shows you how to do it in a synthetic way. I will come to the conclusion. And I think that this talk exemplifies how we can assemble nanomaterials and biomaterials as hybrid systems on electrode, and how we can use this field for bioelectrochemical applications. But I think that we have to look for the challenges and what we should do in the future. Now, I will just mention that from a basic point of view, there is one big challenge, and I think that we try to address it these days by trying to make out of equilibrium bioelectrochemical reactions on surfaces. This is basic science. But from a practical application, I think that we have here a lot of possibilities, and this possibilities relate to energy conversion, making new photobiofuel cells, new biomimetic fuel cells, and particularly to develop, this is something very important from my point of view, biomedical active electrodes for the controlled electrical release of drugs. So with this, I think that I finish. I will thank my coworkers that without them I couldn't do anything. And these are really the future scientists. I only have to give the talks. Thank you very much.